I'd like to introduce you to our two speakers today, Rachel Laliakos and Eric Thierien. Rachel is a medicinal chemist in Schrodinger's therapeutics group, which she joined in 2018. She currently leads a talented team of multidisciplinary scientists dedicated to discovering drugs for neurodegenerative diseases. And she completed her PhD in synthetic organic chemistry at Monash University and began her medchem career at Cancer Therapeutics in Melbourne, Australia in 2012. And Eric is a director of computational chemistry in Schrodinger's therapeutics group, leading efforts in collaborative and internal drug discovery programs with a focus on applying free energy perturbation methods. And he obtained his PhD in organic chemistry from the University of Montreal and conducted postdoctoral research at McGill University in computational chemistry. So I will pass it off to our speakers.
Wonderful. Thank you, Katie. And thanks everyone for joining today. Eric and I are really excited to tell you about the discovery of Chi-11101, our potent selective and brain-penetrant DLK inhibitor. So let's start by talking a little bit about DLK biology. So DLK, dual leucine zipper kinase, is a member of the MLK family and expressed predominantly in neurons. It regulates neuronal apoptosis and axon degeneration in response to cellular stress via the MAP-JNK pathway. You can see that illustrated in the scheme on the right. So in response to neuronal injury or cellular stress, DLK dimerizes, it autophosphorylates, then it phosphorylates these downstream markers, MKK4 and MKK7, and activates the JNK pathway. So it has been found that genetic deletion or pharmacological inhibition of DLK results in reduced synapse loss, neuronal degeneration, and functional decline in models of both Alzheimer's disease and ALS. And so given ALS pathology is driven by a loss of motor neurons, and it's hypothesized that DLK inhibition preserves motor neurons by stabilizing these neuronal survival signals, it makes DLK an attractive target for the treatment of neurodegenerative diseases such as ALS. And so the project goal going into this was to develop a best-in-class, orally available CNS penetrant small molecule DLK inhibitor. How we work, next slide please. How we work here at Schrodinger deserves a little bit of a moment to tell you about the difference in our approach. Obviously here, we incorporate in silico physics-based methods into drug discovery. And we do this quite uniquely by working medicinal chemists like myself and computational chemists such as Eric, working really quite integrally together to solve challenges as they come up. And we develop or have developed, we have an arsenal of models, but we can also develop models as challenges come up to solve a multitude of problems that come at us. So we have models to predict potency, to predict selectivity, on the ADME side, solubility, permeability, metabolism, and then more on the safety side, CYP inhibition, hERG inhibition, brain penetration. You'll hear how we've implemented the models in the program that we're going to describe to you today. Now we'll just start by just talking you through one of the workhorse assays that we use, so we're all on the same page when we start the presentation. So free energy perturbations, or FEP plus, is really our bread and butter. And so the scheme on the right illustrates how it works. So the vertical path in this illustration is the process by which one ligand goes into the binding pocket. And that's really what you want to know the energy of. But it's a pretty complex calculation. And if the calculation is complex, it's probably going to be computationally expensive and time prohibitive. So though we want to know the vertical path, what we could do instead is look at the horizontal paths, which illustrate the free energy transformation of one ligand to another, both in solvent and in complex protein. And since free energy is a thermodynamic property, we can show that the free energy difference of the two horizontal processes is equal to the free energy difference of the two vertical processes. And what this calculation gives us is the relative binding free energy between two ligands. So as medicinal chemists, what do we care about? We care about, is my next compound going to be more or less potent than my current one? If it's more potent, we make it. If it's predicted to be less potent, we go back and we ideate some more. So you can see why having a tool like this would be really powerful when you're starting the discovery process and you're learning about SAR and you're wanting to identify hits, and you really want to hone in on the compounds that are going to be most potent, allowing you to really focus your attention on compounds that are going to advance the program forward quickly. So now let's show you how we've taken these innovative tools and technologies and we've applied them towards the development of a novel DLK inhibitor.
All right, so the starting point of the project was the publication from Genentech of a small chemical series around DLK and also the publicly available crystal structure as you see here on the left-hand side. So typical type 1 kinase inhibitor exemplified by an aminopyridine hinge, a small pyrazole core with two different set of R groups. One is the isopropyl shown here that packs under the P loop, and another piperidine oxytenyl group that is occupying the solvent exposed area. So our strategy relied heavily on FEP, and in order to do so, all that we did was to take the minimal binding motifs, such as the hinge binder shown here, the trifluoromethyl aminopyridine, and we thought of using this as a starting point to enumerate potential cores and then grow back the molecule in order to keep the amenability to FEP. So through core enumerations, we did find new novel cores. These were unsubstituted, and because the next step would be to do and perform some R groups enumeration on any position available on the core that would lead to a more elaborated molecule. And by iterative processes, modifying more R groups and also changing the hinge, then it could lead to a new chemical series. So I'm going to show you exactly how we proceeded in all the steps here. So through the core library enumeration, we generated over 2,000 molecules, and then using docking to eliminate compounds that would clash severely with the protein, we almost took all the library to what we call fast single-edge FEP, which is a very efficient manner to triage a large number of molecules using FEP. And from those that looked very good, we took 47 out of them to run the more accurate prediction of binding affinities using FEP. Out of those 47 molecules that were predicted, we synthesized 23 that were synthetically tractable. Out of these 23, 7 hits were identified with pretty good potency in the biochemical assay, ranging from 50 to a little less than 500 nanomolar. Those were having a very good ligand efficiency, as you see here, and fortunately, there was a quite good agreement between the potency determined experimentally in the FEP predictions. So taking those top hits and looking at their different ways to actually be able to diversify those scores, what we decided to do as the next step is just to perform what we call in silico SAR. So for each of the cores, we enumerated R groups on all the potential position on the core. And what you see on the left is the black dots represent the FEP predictions in the unit of decay called per mole, so delta G of binding, compare with the unsubstituted parent molecule, the parent core, highlighted with the red dot. So for most of those, you see that it was possible to at least see some improvement in the predicted potency, and that influenced the choice and the selection of the cores that we would take forward for synthesis. And this is what we've done with 3 of them. And as you can see, in all the 3 different series, FEP was in a very good agreement, and then we could easily optimize further the potency and while maintaining a very good ligand efficiency. And we ended up selecting the imidazopyridine core as the lead series, the hit series at that time for further identification. At that time, we also wanted to evaluate alternative hinge binders. A very similar approach was taken, leveraging FEP again. So we did a systematic enumeration of the representative hinge taken from literature and publications, and we did the enumeration on R, one of the molecules that we identified before. So although FEP predicted that few of the new hinge would improve potency, we only selected that trifluoroisoindole compound for synthesis, and we showed that the potency was actually tracking well with FEP, although it was at the expense of lower solubility and increased lipophilicity. So we decided to revert back to that trifluoromethylamidopyridine hinge because it was showing a better balance between potency and the properties. So this was used in further optimizing our lead series.
So we rapidly got to this imidazopyridine lead, Chi3540. You can see it's a pretty small, almost fragment-like, really potent. You can see the strong correlation between experimental potency and FEP predicted potency. So again, having having developed that confidence in the predictions at this point really gives us a strong advantage here. So it's potent, it's ligand efficient, its cell potency needs a little bit of work, but it's still a really great starting point to be at. And so with this, we enabled a crystal structure, which you can see on the left. And what that allowed us to do was, of course, crystal structures help enable our modeling further. They show the conformation of the ligand. And it also pointed out clear opportunities for us to grow in terms of achieving more potency and maybe vectors that could be used for fine-tuning phys-chem properties. And so having that available at our disposal, plus a really strong working FEP model, you can see that from Chi3540 to the compound on the right, Chi3723, the methylpiperazine, we modeled about 200 compounds, but we only made about 10. So you can very quickly see the advantage. And now you have a compound like 3723, which is incredibly potent. You can see it's picomolar in our biochemical assay. Its cell potency is about 20 nanomolar. It's soluble, it's stable, it's permeable, really beautiful bioavailability, and also a really workable KPUU, because remember, after all, we still have to get into the brain with these compounds. But we're really excited with 3723 until, you always have that until, we found that it was a really strong hERG inhibitor. And of course, we can't move forward with a compound like that. So we then had to work out how we were going to solve this problem. Now, there are many common ways to reduce hERG inhibition. And of course, we employed those medchem strategies to try to overcome this challenge. So the first thing that you can do is you can try to reduce the log D of the compound by adding polarity. You can see that exemplified with Chi5458, this methyl ether. We reduced log D. We got a slight movement in the improvement of our hERG IC50, but really not significant enough to remove this as a liability. Another common approach is to modulate pKa. Now, a tool that we have at our disposal is Jaguar pKa predictions. And so the first thing you want to do, we would design compounds, we would predict their pKa, and we would see if we're moving the compounds in the right direction. But of course, you want to validate, all models you want to validate with experimental data instead of just trusting them blindly. So you can see here the correlation between the Jaguar predicted pKa and our experimentally derived values are pretty close. All right, so then we have the confidence that we can use those predictions to drive decision making. And what you see now on the right is you see the correlation in this correlation plot between our Jaguar predicted pKa and our measured hERG PIC50s. And we found that we want to be down in the bottom left quadrant. So by synthesizing compounds with a Jaguar pKa of less than 6.5, you can see we significantly reduced our hERG liability. And that's valuable to us because then in our design workflow goes our FEP on target predictions and then our Jaguar pKa predictions, in addition to the other models that we run. And that largely abrogated this problem for us. And so we found Chi 9440, this asymmetric morpholine, and it was really attractive. Again, a really potent compound. You can see the biochemical potency there, still in the picomolar. You can see the correlation with FEP predictions, really tight, solid cell potency. Again, it's still soluble, it's stable, it's permeable, it's bioavailable, still workable KPUU, but we've now abrogated that hERG issue. And so we moved this forward into our ex vivo mouse dorsal root ganglion neuronal degeneration model. And up on the top, you can see the untreated control, and then you can see the addition of paclitaxel induces dorsal root ganglion fragmentation after about 3 days. And now we treat with our compounds. So our DLK inhibitor, 9440, shows, as you can see, a dose-dependent protection from this paclitaxel-induced axon fragmentation. So again, really excited, these ebbs and flows. And you know what happens when you're really excited? We found, of course, that this compound was a really strong CYP3A4 time-dependent inhibitor. And of course, we had to fix that. So what we then did is, of course, through MedID studies, GSH trapping experiments, we try to identify where on the molecule are our liabilities, our sites of metabolism. So we identified 3, the morpholine region, the ethyl imidazole portion, and the hinge aminopyridine. And so we set about to systematically block as many positions as we could to try to offset that 3A4 time-dependent inhibition. And so we started on the morpholine. We systematically blocked every site. They were all still TDI positive. We then tried to replace the morpholine entirely, still TDI positive. We tried to sterically hinder the morpholine by substituting the 7 position of the bicyclic ring. Again, that did not work. We tried then to move to block the imidazole methide formation down on the imidazole ring, and that did actually work by blocking by removing that alpha proton. You could no longer form that reactive metabolite, but that came at the loss of potency. And so it wasn't something that we were willing to offset. And so at this point, we really had worked this series quite extensively to try to reduce this TDI. And what we went back and looked at is, can we change the electronics of the core? And what you'll see is that actually was our solution here. So on the next slide, you'll see the value of having FEP to guide our synthetic choices is that you don't usually want to go back and change your core, because obviously it's almost like taking a step backwards, right? You're stepping back from your lead series, and you kind of have to start again. Though you've learned a lot of SAR from your parent series that can be transferable, it is a step backwards. And of course, you have to then change all of the chemistry to enable this new core synthesis. But the value of having FEP predictions to kind of guide those core choices at least helps you feel confident in the synthetic investment you're about to make. So we have about 5 different cores with different heterocyclic cores exemplified here. The first point is you can see the strong correlation, again, between the FEP potency and the experimentally derived potency. So again, we were able to make compounds that were potent. And by changing these electronics, we ended up with 2 different ring systems that did not show the CYP3A4 time-dependent inhibition. One was the indazole, one was the pyrazolo-pyridinone series. And based on all of the overall properties, we don't have all the properties here today, but the pyrazolo-pyridinone series provided an overall balanced profile and was prioritized for further optimization. Now, within that core system, there were examples where the TDI started to pop back up. And we did not want to relive the experience that we just had with the imidazole-pyridine series, where we did everything so slowly and systematically. And we thought about, is there a smarter way that we can add some of our computational models into this process to help us make better decisions, so we did not repeat the mistakes that we made with the mistakes and the time that we spent on that imidazole-pyridine series. So I'll tell you about the QM approach that we enabled.
Yeah, so basically, based on the MET-ID studies that we perform on representative molecules of that series, we found that there was a reactive imine formation from the hinge aminopyridine motif that was actually leading to potentially that observed time-dependent inhibition. So we developed a very simple QM-based method where we look at the imine species here, that cationic imine species, and we use QM to calculate the energy in terms of saying if it's stabilizing or destabilizing that intermediate that is reactive. So from what you see on the plot, this is based on retrospective analysis, and when we developed the method, you see that the compounds that had the higher TDI change were actually having the more stable, lower energy from QM. And the compounds that we're interested in pursuing that had a much higher energy would destabilize the imine formation and hence be less TDI active. So basically, using that method, we were able to discriminate between identify that the OCF3 series was actually a little less problematic than the OCF2 series. Although applying the method, we saw that we could easily rescue the OCF2 series by adding a proper substitution on the bicyclic system, such as that trifluoroethyl group, where the energy of the imine formation was suggesting that it would be less of a problem in terms of TDI risk, and that was indeed the case. So that method was very simple, not relying on the binding to any of the CYP, but that was purely a ligand-based approach and really focused on that specific area of the molecule. But that was kind of helpful and instrumental for us to actually understand the mechanism and de-risk a lot of ideas. So another challenge that we were facing, so Rachel mentioned about KPUU as a brain penetration metric that we measure, but these are expensive experiment that needs to be run in vivo. So although we had a look at in vitro assays such as the permeability, the MDR1 cell line, we thought that we would need a better tool to guide chemistry decision and trying to further optimize brain penetration. Using commonly known metrics available to us, we didn't see any correlation with our chemical series, unfortunately. And even just lipophilicity itself was not really guiding us in getting a KPUU of 0.3 or above, as it is exemplified here in the plot in the green section. So we developed what we call the QM-based approach to calculate the energy of solvation, namely ESOL. And that's a property that is actually calculating the coefficient of the molecule between water and gas using an implicit water model. And that actually factors in the 3D shape of the molecule, the size effect, the polarity, and also we have an equation shown here below that state penalty whenever you work with molecules that have tetrahedral groups. So using that method, you see here on the plot that from ESOL correlation with KPUU that we had a pretty good correlation, and using a value around -17 was guiding us in terms of optimizing our probability to get a compound with decent brain penetration. So all of those findings are actually published this year in GCI. Just to highlight a little bit of examples from the molecules that we work at that time using ESOL. These are all substitution on the sub-unexposed group where we can modulate ADME properties. And if you look at the measure or calculated properties that we have here, only ESOL was actually quite accurate in predicting compounds having greater KPUU, and exemplified with some very simple molecules, but then sometimes a small change would be detrimental to brain penetration, and it was well captured by ESOL. Another challenge that we faced, this is a kinase inhibitor for brain penetration and neurodegenerative disease, so we need to be very selective against other kinases. And one representative compound from our lead series, Chi-10634, we ran a scan max and trying to identify potential off targets, which we show here, and some of them had quite potent inhibition against them. And the goal was to try to improve the selectivity fold over DLK in greater than 50. So using our FEP and available crystal structure or homology models, whenever crystal structure was not possible, we developed FEP models for most of those off targets where we can run prospectively and try to optimize selectivity profile for any of the design molecules even before we need to synthesize them. So we also implemented that approach into a selectivity MPO function where we can rank order compounds based on their selectivity profile as well. In this specific case here, although we had 6 off-target that we identified that were the key ones to dial out, we found that there was a pretty good correlation between TRK, KIT, and FLT3, so we use FLT3 as a surrogate. And what you see from the 4 FEP models that we used throughout the project, that the through perspective performance highlighted the correlation plot that you see here comparing FEP predictions and experimental binding potency for all of them. Performance from FEP was actually very, very good. So using that selectivity FEP tool and models, we were able to identify an optimized molecule where we could improve selectivity. So starting from 10634, where the selectivity profile was not so great in our selectivity MPO, highlighted by a very low score, we found that introducing a chiral center in that 6-member ring here in a specific cis conformation would improve selectivity, and that was confirmed by having, run scan max and looking at very high selectivity profile now for that molecule. And we also got a crystal structure of a relative compound here, where you show that the cis configuration of the THP ring with position the methoxy up the roof and interacting with those 2 residues of valine 131 and phenylalanine 192 in a way that was optimal for selectivity. And it's important to know that those 2 residues here have very low conservation score across the different other kinases that we looked at, and that would explain the greater selectivity against all of them and favoring DLK in this case. So all the tools that we developed to help chemists were included in that in silico workflow during lead optimization stage. And we tried to use a model to predict almost all the endpoints that were important for us to guide chemistry decision. So basically, the workflow would start as first all the ligand-based QM approaches, ESOL for brain penetration, log-D that we predicted using QM, so make sure that we have a very good prediction and accuracy in predicting log-D and lipophilicity, because we haven't mentioned it, but we found pretty strong correlation of log D with FG2 cytotoxicity. Again, PK was important to mitigate hERG issues, and also we use a QM method to mitigate any potential TDI risk. All the compound that satisfied those cutoffs here, we actually ran FEP on DLK to get a very good prediction on the potency. And all the compound that would survive the first step, the first and the second step would go through our selectivity profiling using FEP and off-targets that we identified. All those metrics would be captured in Live Design, where it's a collaborative tool that can be accessed online, and making sure that all the chemists and people in the team would be able to collaborate and visualize the result and make sure that you can take good decision in selecting the compound to proceed for synthesis. So having that computational workflow developed, we're now going to show you how we proceeded with the lead series and the discovery of Chi-1111, obviously.
Yeah. So Chi-1111 had really attractive properties, and we wanted to understand that now in our ex vivo and in vivo models. So we started, of course, with these ex vivo assays, and we're able to show that Chi-1111 blocks DLK activity and modulates levels of translational and disease phenotype in ex vivo assays. So it's probably important at this point to note that, Eric mentioned that Genentech had already initiated a DLK program. They were in the clinic at this point, and they published the structure of their clinical asset, GDC 0134. And so what that enabled us to do was to have a strong compound to benchmark against. And so in our 3 assays, in our target engagement assay, where you see paclitaxel-induced phosphosphere Jun levels, and in our phenotypic protection assays, where you see paclitaxel-induced axon degeneration, and where you see neuronal growth factor withdrawal-induced axon degeneration, comparatively, you can see that we performed quite well, if not slightly better, than the Genentech compound, showing stronger inhibition and stronger effects across, the biochemical, cell potency, target engagement, and these phenotypic assays. So that was really gratifying for us to know that we had a really strong competitive compound here. We then moved this compound in vivo. Another company that was working in this space was out of MD Anderson, Magnolia Neuroscience, and they actually published a paper that demonstrated that DLK is constitutively active in the cerebellum of healthy mice, and so thus we could measure DLK inhibition directly in this non-injury model. So it was nice to be able to leverage the literature again here, and what we can see that in this PD model, we have linear increase of brain exposure in the plot on the left, and then in the center we can see that dose-dependent phospho-C-Jun reduction. We quantified it, normalized it to total protein level, and on the plot on the right we could calculate a cerebellum phospho-C-Jun IC50, which gratifyingly, when you apply all the corrections to get your unbound concentrations, correlates perfectly with our in vitro HEC-293 based phospho-junk IC50 assay. So that was a good position. We then moved this compound through preclinical PK and in vitro safety. You can see from the plot that we demonstrated desirable oral PK profiles across rodent and non-rodent species. If you have a sharp eye, you will see that disconnect with the dog. Our dog clearance was underpredicted by both microsomes and hepatocytes, suggesting there's some sort of extra hepatic clearance mechanism involved. We ran in vitro med-ID from hepatocytes. We ran med-ID with in vivo samples and identified a dog-specific metabolite, but more studies would be needed if we want to further understand the clearance mechanism in dog and how it would impact our human clearance predictions. Now, in terms of the safety, you can see we have a pretty reasonable safety profile. We abrogated our HERG issues. The compound is not cytotoxic or myotoxic. Eric spoke you through our selectivity process such that we ended up with a pretty attractive profile here. Of course, the only one to mention is LZK, which is a close homologue of DLK. The ratio is about 24-fold, so not as high as we wanted it to be, but given the high sequence homology between those two homologues, it's probably as strong of a offset that you're going to get. We were clean in our safety 87 panel. We had a low DDI risk. You can see the predicted human dose was at 80 to 250 mg once a day to cover phospho-junk IC50 at trough, and we knew from the literature that trough coverage over the dosing period was shown to be necessary to achieve neuroprotection in 2 different efficacy models. And so Chi-111 really was a promising preclinical candidate that we identified through our drug discovery efforts. And so in summary, we've talked you through today program initiation, from how we identified hits to the trials and tribulations of any drug discovery program. All of the models that we threw at those challenges to help us pave a way forward. You can see how the predictive DLK FEP binding models, the plot is on the bottom left, that strong correlation really allowed us to make solely potent compounds, and that really is a tremendous advantage when you're running a drug discovery program. We only had to synthesize less than 1,000 compounds, and we evaluated 30,000. So, our ideas were broad, but we only had to synthesize a very small subset of those. Throughout the program, we utilized models that were existing, but we also established new models as challenges arose. For example, you heard about our QM-based ESOL method for predicting KPUU really significantly impacted the program favorably by increasing the probability of identifying brain-penetrant DLK inhibitors. And of course, this is now an integral workhorse of all of our neuro programs that we have run since. So a really powerful tool there. And then we've also talked you through some of the multiple common medchem challenges that we overcame. HERG, CYP3A4 TDI, you heard about the QM method. We didn't talk about the cytotoxicity today, but you heard about our approach towards kinase selectivity. So, we hope Eric and I have shown you through this webinar the power of integrating in silico tools into the discovery process. Think about adopting a predict first mentality. It can only help you and enable you to make better decisions faster. And so with that, we'll go to the acknowledgements. A really strong program team helped us get to this stage. Our project leader was Zini. This program was in collaboration with Takeda. We delivered the tool molecule. There are some assays that they ran internally, late-stage assays that hopefully we will be able to publish or present on at some time in the future, but we thank our collaborators at Takeda. And of course, the project team and everyone here for listening today. So we welcome any questions that you might have and hope you enjoyed the talk.
Awesome. Great. Thanks so much, Eric and Rachel. We have a lot of really good questions coming in. So let's dig right into them. Starting from earlier on in the presentation, looking at the novel core search selection generation, does it require protein structured information, or what information did you use to search the cores? And then similar question, how did you generate enumerations from the core structures?
All right, I can take that question. Yeah, so basically the way that we proceeded was to use the enumeration tools that we have in Maestro and looking at regions or just bicyclic systems that are unsubstituted, 5-5, 6-5, 6-6, and even monocyclic systems in a very pure systematic way just to enumerate. And then we use docking to select those that would satisfy the binding pose. Obviously, with a very small core, most of them, like I show in the funnel approach, we're satisfying docking, and then we use those docking pose to proceed to FEP, where we have a much better assessment of their potential binding affinities.
Great. And please feel free to continue to submit questions as we go through these. I will say here we've got a couple questions on whether this will be recorded. The talk will be recorded and available, and you'll be sent a link after the talk to access that. Okay. A question around selectivity. You later in the talk then went through the FEP selectivity, but when you were looking at the new core combinations in the beginning, were you accounting for selectivity then, and how did you account for selectivity during the evaluation of the new cores?
Yeah, that's a great question. I think, like Rachel mentioned, predict first mentality is important. Although at that stage, without knowing exactly what are going to be the off targets from the kinase domain, it would be very difficult to assess selectivity up front. So basically what we've done is we secure a new core series, having multiple cores. So if something wrong happened, and like we showed, it did, so we had to go back, and we didn't have to go back to hit ID. We already had a backup core series that we could easily rescue when needed. So basically the selectivity came later once we had advanced enough molecule in the series to run the scan max and identify the off targets. But yeah, you could leverage it before. It's just it might be a little more difficult to identify which one unless you want to run selectivity profiling against the entire kinase. But that might be a little bit too much.
Okay. There was a question, when you did the crystal structure, did you mention that you had some ideas on how to work on it? It seems you implied that your model wasn't showing all the details you observed in the crystal structure. What details were we able to observe that weren't present in your model?
Yeah, I can take this one as well. So the way that we use the stepwise approach was to enumerate cores, and for those cores that were not purely symmetrical, there was potentially at least two conformation where the binding pose could adapt. And given that, and using FEP, although sampling is happening, but once you start growing the molecule too big, you would lose the capability of sampling across that specific torsion, and the two conformation of that specific core would be important as a starting pose. So basically, the crystal structure at that time was instrumental in guiding us on what conformation was preferred in the binding mode, so we could use it to further explore the different vectors, and then use that as an input pose for FEP to mitigate any potential mispredictions.
Great. Okay. There was a question on a specific slide. So the basic group, like methyl piperidine, is not necessary for binding. It's just a soluble group.
Yeah, that's correct. That's correct. That region, we didn't get a lot of potency from that region. There was a bit of potency to be gained, but it really was solvent exposed, so we could tune the physical chemical properties of the molecule in that region, and you can see as the compounds evolved where we really were able to leverage that as a handle.
Okay. question. As you discovered more exclusions, such as the core with the CYP liability and your metabolism blocking sites, how did you manage those as part of your iterations through computational models? Typically it gets harder than adding more computational filters like pKa or ESOL.
Yeah, I can take this one. Definitely, that's a challenge to have more and more models, but the way that we include all the models into, the selectivity, not the selectivity, but the NPO scoring functions that we had in my design, and making all the endpoints, the calculated endpoints available, was very good in triaging large, set of designs and ideas to focus only on those that had the best probability to succeed and satisfying all the different models that we have developed.
All right. Lots of good questions coming in. Great work. Can you please elaborate on your MedChem-ComChem collaboration process? Were only compounds made that were based on FEP predictions were favorable?
Yeah, that's a good one. That's a really good one. I guess, it depends. Eric and I work really well together, so that helps, right? We had a really strong team, and I think kind of the ethos for medicinal chemists working at Schrodinger is a willingness to really work hand in hand with the computational chemistry team. And it's probably kind of a mentality that could use some more adoption external to Schrodinger as well. But everything is done in tandem as a team. We're designing ideas, we're running the predictions, we're looking through the cycles together, we're making our decisions together. You asked, did we only make compounds that were predicted to be good? That's a really good question. At the start of a program, you probably want to be a little more exploratory, because you have not, medicinal chemists are skeptical people. You want to make sure that the model is robust and performing well. So maybe earlier, before you've established confidence, you might make compounds that are predicted well, that are predicted to be okay, and are predicted to be poor, just to make sure that the performance of the model is strong. But as you start to develop this series, and as your model, you keep iterating, you make more compounds, the model keeps getting better as you move along. And so it would be foolish with a well-performing model to not follow the predictions once you've established that strong correlation between the predictions and the experimental potency, as you saw we showed throughout the program. Anything from your perspective, Eric?
No, I think it summarizes very well. If I can just add in detail, you're right. I mean, at the point where we develop models, the computational chemist might come to the chemist and ask for very specific compounds to address an hypothesis on the model, to validate the model further, even if the compound doesn't have any potential of becoming an interesting compound in the medchem perspective. But these are very important for us to develop more robust models. And like Rachel mentioned, working hand to hand together and having the same philosophy on how to leverage modeling tools and helping chemistry, I think this is unique and something that people need to start thinking about a little more in leveraging all the capabilities that the software can offer.
Yeah, and then maybe one further thought on that, having that good relationship between the computational chemist and the medicinal chemist. Of course, Eric might come and say, I need this compound, to enable a reference for a full cycle closure. Can you synthesize it for me? And as a chemist, we might look at it and say, this is really challenging to make. Is there an alternative that's more synthetically tractable that will still serve your purpose, that'll be easier for us to synthesize and still gives you what you need? So again, it's really about establishing that relationship, understanding what both sides need to move their functionalities forward is really the key to success in integrating these tools.
Great. Okay, question that out of curiosity, between Chi 11101 and Genentech's clinical candidate, the similar portion is the amino pyridine, but that was not patented, question mark?
No. So, well, of course, the sharp eye, good comment. Eric talked you through one of these slides. The hinge stayed consistent, and we did maintain that. And we started with it, as we said, because it was small, ligand deficient, good starting point. Now, we tried to move away from that. You saw the workflow that we did, but it ended up being the most efficient and effective hinge binder. So we maintained it. But that is the singular part of the molecule that remains the same. The whole southern portion, the cores, the solvent channels are all significantly different. And so if you think about it, in terms of perhaps a Tanimoto similarity score, they are significantly different from a patentability score, but from a patentability perspective, we do have patent coverage. So no, those were not exemplified. Our core ring systems were not exemplified previously. And so, yeah, it's a good observation and a good question. But our compounds are significantly different outside of that core.
Okay, a couple questions coming in on this. Curious, the synthesis and how many compounds, do you know how many compounds were made to sort of get to Chi-111 following these in silico approaches?
Yeah, so we mentioned we made just south of 900 compounds, which if you think about a traditional program, which could be upwards of thousands, it is significantly reduced. Now, if you think about the things that we did that we probably could do better, that TDI challenge on the imidazopyridine series really kind of hung us up. And you saw we went through a very traditional and systematic approach to block all those sites of metabolism. That took us quite a bit of time, and we had to make a lot of compounds. And what we realized through that was we could have done this better, and we could have done this smarter. And a lot of this program exemplified for us learning opportunities for future Schrodinger pipeline to do things smarter and more efficient. And so now, the QM-based approach for thinking about your reactive metabolites, and can we use that to abrogate the issues, or ESOL for brain penetration. All of these things will allow us moving forward, and you'll hear more stories from Schrodinger where we are making significantly less compounds. But even so, 900 compounds is a pretty respectably low amount of compounds to get to where we were, especially for a CNS program where the bar is so much higher in terms of safety and selectivity profile for a chronically dosed CNS agent. And so, yeah, we're pretty proud of where we landed with this program.
Great, sorry about that. So a question around the difference between docking and FEP. So it says normally docking is not recommended for a rank ordering of compounds. Is this sort of the strength of FEP? And maybe you guys can explain how you think about the difference between docking and FEP.
Yeah, that's a good question. So docking, has been used for several years now, and FEP is more recent, I would say, and definitely more compute intensive in regards to time of calculation. But technically, docking, the way that we use it here is not to rank order compounds at all, because as you can see by yourself or look at some of our recent studies, is the scoring function from GLIDE, it doesn't always correlate well with binding affinity, and it has some limitations. So the way that we leverage docking in our program here for the DLK program was basically just to get a starting post for FEP, or eliminate ideas and designs that would be such that they would have a severe clash with the protein, and hence be not a good binder to the structure itself. But most of the time it would just take everything to FEP, which prove, as you see on the plot on the bottom left here, that the prospective performance was quite good And even at some point, I had a chemist, the project leader, the chemist, saying that why do we still need to run the biochemical assay, because we can trust FEP is so good. So that's maybe not an exception, but that's a case where FEP performs very, very well throughout the project, which is not always the case. But basically, whenever you can, FEP is very, very good at predicting binding affinity, much better compared to simple docking using Glide.
Right. Along with the physics-based approaches, did the team tried, or has the team tried any ML models for HERG or CYP3A4 property predictions?
Not at that time. You need to understand that to build a good machine learning model, you need to have enough experimental data. So basically, as soon as you generate enough data, you might be able to apply and develop a machine learning model to see and observe some trend, but very limited data for such expansive experiment. It's not always amenable to. And using more global models based on very diverse chemical matter is usually much less accurate. So we decided to rely more on physics-based approaches, such as the QM methods that we mentioned today.
Yeah, and I guess to add on that, of course, as Schrodinger's internal pipeline continues to grow and we develop and we collect all of this internal data, we can integrate that more in our newer programs, right? Because as Eric said, you need data to base the model on, and we don't have the luxury of perhaps a big pharma who has decades and decades of data to leverage, but we are using machine learning much more integrally in our later programs that you will hear about at some point in the future.
Great. A very inspiring talk. Thank you. Is the ESOL method high throughput? How many compounds can be calculated at a time?
It's a QM-based method, so this is definitely not super high throughput, but if you have access to multiple CPUs, because these are running on CPUs and not GPUs as opposed to FEP, but if you have access to multiple CPUs and run in parallel, you can easily scale it up to a certain amount of compounds. As you see here, we have actually profiled over 17,000 ideas throughout the project. And routinely, we would, run profile compounds with ESOL on 100 and 200 number per day, given our throughput. So, and more recently, we have implemented ESOL in live design. So these are very easy to run directly in live design, and usually you can get result overnight if you submit the night before. So it's, again, time consuming a little bit. It's compute intensive, but this is not limiting the use of ESOL to actually profile prospectively significant number of molecules.
Yeah, and maybe to add on that point, if you do have access to live design, for example, me as a medicinal chemist, I can draw a set of ideas. Maybe I'll draw 10, maybe I'll draw 100 ideas, and now I can just click to run them, and I'll have the results in the morning. So, earlier you had to work closely with the computational chemist. Now it's a little more enabling if you have access to live design, and of course more throughput in that way because it's just click to run.
Great. One more question here, maybe one more. Did you use models, and what models were used for new ideas to see if they were synthetically feasible or not?
Well, that's the value of having a synthetic organic chemist on the team, right? And that's why this works so well, because we do a lot of hand draw design. So of course, whatever we draw is very likely to be synthetically enabled. But we do a lot of enumeration work, auto designer work, where we bring in synthetically tractable routes to design enumerated compound libraries. But again, at the end of the day, whatever we select is you have a visual inspection by a synthetic organic chemist who is trained on how to make molecules. So kind of having that skill set within the team is really quite important. But we have other tools, right, Eric, on predicting synthetic tractability. We have scores that we've generated, but that's probably more appropriate in cases where you don't have a chemist accessible to you, or if you just want to triage large idea sets to ones that are predicted to be more synthetically enabled, because you don't want to weigh your synthesis queue down with really challenging compounds unless they're really quite unique or interesting or have really strong predictions behind them.
Great. All right. Well, we are officially over time, and thank you both so much, Eric and Rachel. Really fantastic presentation, and thank you all for joining us. Have a good rest of your day.
Thank you all. Reach out if you have any questions.